I would like to introduce now the next speaker, who is Stephanie Longet from University of Oxford. We had a chance to interact by email, so I'm so happy to have her here. She will be talking on COVID-19 mRNA vaccination, driving differential mucosal neutralizing antibody profiles in naive and SARS-CoV-2 previously infected individuals. Welcome, Stephanie.
Thanks for the introduction and the invitation. So I'm going to share my screen here. And oops. Yeah, and I'm going to use a pointer. Okay, so good morning or good afternoon. So I'm currently postdoctoral researcher at the University of Oxford in the UK, and today I'm going to speak about mucosal antibody responses in naive and SARS-CoV-2 pre-infected individuals. And all data I'm going to show you today were published last year in Frontiers in Immunology. Okay, so as you know, SARS-CoV-2 is transmitted by the airborne route. So mucosal immunity is very important to protect against infection. So the goal of this study was to characterize antibody responses in oral fluid, which mirrors mucosal responses after COVID-19 mRNA vaccination. To analyze the antibody responses, we use two mesoscale discovery multiplex immunoassays. To analyze the binding of antibodies, we use an indirect ELISA, but here in each well, you have 9 antigens, SARS-CoV-2 spike, RBD, and SARS-CoV-1 and MERS spike antigen, and spike antigen of the seasonal coronaviruses. And basically here we use a secondary antibody, which was sulfotag. It was either an anti-IgG or an anti-IgA. To analyze the neutralizing responses, we use a surrogate neutralization test, an ACE2 inhibition assay, and basically the same principle than the indirect ELISA, but here in each well you have a range of spike variant antigens, and basically it's a competition assay between the antibodies in your samples and ACE2, which was sulfotag. And we use a mesoscale discovery reader to read both plates. Okay, so the specific aim of the study. The first aim was to evaluate the sensitivity and specificity of the multiplex MSD ELISA to detect SARS-CoV-2 specific IgG responses in oral fluids versus plasma. The second aim was to compare the kinetics of SARS-CoV-2 specific IgG responses induced by the Pfizer-BioNTech vaccine in oral fluid and plasma side by side. And the third aim was to evaluate the impact of a previous SARS-CoV-2 infection on vaccine-induced mucosal IgG and IgA responses, and also neutralizing responses following mRNA vaccination. Our cohort was composed of 200 UK healthcare workers. All of them were based in Sheffield in the UK. Some of them were SARS-CoV-2 pre-infected, and others were naive to SARS-CoV-2. We collected oral fluid and plasma before vaccination, 28 days following the first dose and 28 days following the second dose. For a subset of participants, we also collected oral fluid 28 days following a third dose of mRNA vaccine. Okay, so now I'm going to show you some key results, and if you would like to know more about this, you can read the paper to have more details. Okay, so here you can see SARS-CoV-2 spike-specific IgG responses in oral fluid and in plasma side by side. In gray, you have the naive participants, and in red, the pre-infected participants. And you have all time points, before vaccination, post-first dose, post-second dose. You can see that you can see there is a linear increase in spike-specific IgG responses following each vaccine dose in naive participants in both in oral fluid and plasma. In pre-infected participants, there is an increase, especially after the first dose, and a plateau after the second dose in both oral fluid and plasma. We determine a strong correlation between spike-specific IgG responses in oral fluid versus plasma in naive and pre-infected participants. But the sensitivity was lower in oral fluid compared to plasma, while the specificity was similar. As IgA is the most abundant antibody in mucosal fluid, we decided to analyze also spike-specific IgA responses in a subset of oral fluid from naive and pre-infected participants. And you can see that mRNA vaccination can boost IgA responses, but only in pre-infected participants and much less in naive participants. So this results means that mRNA vaccination, so systemic mRNA vaccination, can reactivate IgA responses, but only in pre-infected participants, so only if people were previously infected or at least maybe exposed to SARS-CoV-2. We also determine a moderate correlation between spike-specific IgA and IgG responses in oral fluid from pre-infected individuals. And we determine a moderate but significant correlation between IgA responses in oral fluid and plasma from pre-infected individuals following 2 doses. And actually, both results here suggest that some IgA may come from the blood, similarly to IgG. We analyze the neutralizing responses using an ACE2 inhibition assay. So here you can see SARS-CoV-2 ancestral spike. You can see the result for the SARS-CoV-2 ancestral spike antigens. And basically here we look at the percentage of ACE2 inhibition. So here you can see there is a linear increase in percentage of ACE2 inhibition following each vaccine dose in naive participants. And in pre-infected participants, there is a significant increase after the first dose, but a plateau after the second dose. Here you can see ACE2 inhibition against a range of variant spike antigen in oral fluid following 2 doses. So you have alpha, beta, gamma, and delta, and also the ancestral spike. So here what you can see, if we compare just the global picture of naive and pre-infected individuals, the neutralizing responses in oral fluid are quite similar, and the lowest percentage of ACE2 inhibition was observed against gamma in both naive and pre-infected individuals. Finally, we decided to look at the impact of a third mRNA vaccine dose on antibody responses in oral fluid. So here you can see spike-specific IgG responses in oral fluid. So basically, it's a comparison between the levels after the second dose and levels after the third dose. And you can see that a third dose of mRNA vaccine can boost IgG responses, but only in naive participants and not in pre-infected participants. We also analyze spike-specific IgA responses in oral fluid, and I would say similarly to our observation after the second dose, the third dose also boosts IgA responses, but only in pre-infected participants. And finally, finally, here you can see the ACE2 inhibition against a range of variant spike antigen in oral fluid following the third dose. So again, if we compare the percentage of ACE2 inhibition between naive and pre-infected individuals, the global picture is quite similar, and the lowest percentage of ACE2 inhibition was observed against Omicron BA.1 in both naive and pre-infected individuals. Okay, so to conclude, we observed that vaccine-induced IgG responses strongly correlated in plasma and oral fluid from naive and pre-infected individuals, respectively. Mucosal neutralizing responses were observed in both naive and pre-infected participants following vaccination with a systemic mRNA vaccine. but mucosal IgA responses were more often detected in pre-infected individuals compared to naive participants. So the potential complementary function of mucosal IgG and IgA in pre-infected individuals may be an advantage, but the clinical significance remains to be determined. Okay, so finally, I would like to thank my supervisor, Professor Miles Carroll, and also Professor Tushan De Silva, who co-led this study. And I would like to thank my students who work on this study with me. If you would like to read the paper, here you have the QR code. And just to let you know, tomorrow I will move to France and I will start a new position as assistant professor and lecturer. So yeah, I'm happy to answer your question.
Wow, Stephanie, congratulations for that. And thank you so much for this really interesting talk that sheds light on so much, and just changes the landscape of vaccination science and research. I know we have a couple of questions, so I'll let Shivali ask us. Shivali, you are muted.
Hi, am I coming through now?
Yes.
Sorry about that. Yeah, I just wanted to say, Stephanie, that was a really great talk. Thank you so much for that amazing data. And actually, because I also see that you're in your new position, you're going to be working on TRMs, it seems like. This sort of perfect question for you. I'm wondering, you show all this really great data when it comes to antibody responses and, you know, sort of B-cell mediated responses. I'm wondering how you think these different levels of vaccinations might be inducing T-cell memory at, you know, of course, you have circulating the cell-mediated immunity, but I'm more curious as to what you think is happening at the mucosal level there and how the T-cells might be interacting and boosting that antibody response.
Yeah, it's something I'm going to look at basically in my new project, because we know that breakthrough infection induce TRM, yeah. But for systemic vaccination, if systemic vaccination can reactivate, it's less clear. It's something we have to look at in details, basically.
Yeah, thank you. That's it's very definitely an interesting area to look into with all these respiratory viruses. But thanks so much for clarifying.
If I may ask a question, and sorry, I'm looking up, you're on a big screen here in the conference room. I actually was curious if you have any data on patients who've, you know, had any immunosuppressants, even mild ones like prednisone and so on. What's happening to their response in this case after multiple vaccines?
No, I don't have. I collaborated on a study where we analyzed immune responses in immunosuppressed people. Yeah, I analyzed antibodies in saliva as well. It was the problem, we didn't have a proper control in this study, in collaboration. So it was not too bad actually, but maybe it might depend on which type of immunosuppression. So we didn't run a proper study to analyze that. So I don't have proper data. I cannot really answer your question, but based on the collaborative study, it was not too bad. There was also a boost of IgG responses. I'm going to focus more on IgG because I especially look at IgG. So there is also a boost of IgG in saliva. But yeah, cannot say more about this.
Thank you, and congrats on the position.
Thank you.
So now, if I may, Pavan Raj, I'm going to jump in as well. Great talk, Stephanie. And again, also congratulations on your new position. And I guess now you'll be able to speak much more French, leaving out the UK, going back to France. C'est fantastique. So my question to you is, so in all these studies, elegant studies, you identify the ability of these, the responses, the antibody responses to neutralize in the context of that neutralization. And as Shivali had commented, we know that there's way more to the immune response, the T cell response, the CD8 T cell response. Have you looked in those patients, or those, have you looked to see whether you can correlate directly your levels of IgA and neutralization with actually things like severity of disease or ability to accelerate elimination of virus?
Yeah, no, we didn't do that in this study. In another study, we look at IgA responses after Omicron breakthrough infection, and we try to use machine learning to try to understand finally the parameter which could impact on breakthrough infections. If I remember, yeah, IgA, but I would say SIgA. Here we look at IgA in general, and I think the most important is SIgA, and we can find it might help to protect. And there is also a paper which was published in New England Journal of Medicine. I think they found that SIgA, the presence of SIgA was important for protection. It was significantly important. But it's not the only parameter based on our machine learning study. It's one parameter among other parameters, and yeah, it's not the only one. But yeah, SIgA is important.
Thank you very much.
Thank you once again, Stephanie. I know there's one question in the Q&A, but maybe you can answer that via chat.
I can do it.
But once again, congratulations and all the best. I'm sure you're going to do exceptionally well.
Thank you. Bye.